Hey there. Hope everyone's hanging in there. Alright, O-level biology, lesson 2. We're on cell structure, organization and microscopy. The cell is the smallest unit able to perform the processes of life. Animal and plant cells are eukaryotic. Genetic material is enclosed in a nucleus. Both contain a cell membrane, cytoplasm, ribosomes and mitochondria. DNA holds the code, genes are functional stretches of that code, and chromosomes package it for cell division. Keep that nesting clear. The membrane controls exchange, cytoplasm contains enzymes for metabolic reactions, ribosomes assemble proteins, and aerobic respiration occurs in mitochondria. Read left to right, inputs enter, the living process transforms them, outputs leave. Name what goes in and what comes out before memorizing the word equation. Plant cells additionally have a cellulose wall, a large permanent vacuole and, in photosynthetic tissues, chloroplasts. The wall prevents bursting and supports a turgid cell but is freely permeable. Climb the arrows. Cells build tissues, tissues build organs, organs cooperate in systems, and systems make the organism. Each level adds organization you cannot see at the level below. Prokaryotic bacterial cells are smaller and have no nucleus or membrane-bound mitochondria. Their circular DNA lies free in cytoplasm, and small DNA rings called plasmids may carry useful genes such as antibiotic resistance. A cell membrane lies inside a supporting wall that is not made of cellulose. DNA holds the code, genes are functional stretches of that code, and chromosomes package it for cell division. Keep that nesting clear. Some bacteria have a slime capsule or flagellum. Viruses are not cells. A protein coat encloses genetic material, and replication occurs only after viral information enters a host cell and redirects its machinery. DNA holds the code, genes are functional stretches of that code, and chromosomes package it for cell division. Keep that nesting clear. Structure is related to function in specialized cells. A red blood cell has hemogloHbin, a biconcave shape for large surface area, and no nucleus, leaving more space for hemogloHbin. A sperm has a flagellum, many mitochondria and enzymes in its acrosome. A root hair cell's long projection enlarges its absorbing surface, while palisade cells contain many chloroplasts. Ciliate cells move mucus, and neurones have long fibers for transmitting impulses. An adaptation earns explanation marks only when its effect is connected to the cell's function. Multicellular organisms show levels of organization. Similar specialized cells form a tissue, different tissues form an organ, organs cooperate in an organ system, systems make an organism. Climb the arrows. Cells build tissues, tissues build organs, organs cooperate in systems, and systems make the organism. Each level adds organization you cannot see at the level below. The stomach, for example, contains muscular, glandular, epithelial, blood and nerve tissues. A leaf includes epidermal, mesophyll and vascular tissues. Increasing size lowers surface area to volume ratio, so diffusion across the outer surface becomes insufficient and specialized exchange surfaces and transport systems are required. Light microscopes use visible light and can reveal cells, nuclei, and chloroplasts. Magnification describes enlargement, whereas resolution is the ability to distinguish close points as separate. Greater magnification without greater resolution adds no detail. Magnification equals image size divided by actual size. After units are matched, 1 mm equals 1000 micrometers. Prepare a thin specimen, add a suitable stain, lower a cover slip without trapping air and focus first at low power. Draw clear unshaded lines, correct proportions, and straight non-crossing labels. Convert image and actual size to the same unit before using magnification equals image size actual size. For comparisons, use paired statements about the same feature rather than two unrelated lists. A cell image is 36 mm long and its actual length is 60 micrometers. Convert 36 mm to 36,000 micrometers, then divide by 60. Magnification is x600. Do not say every plant cell has chloroplasts, that bacteria have no DNA, or that a large image must show greater detail. Always include units during size calculations. DNA holds the code, genes are functional stretches of that code, and chromosomes package it for cell division. Keep that nesting clear. Practice. Explain two adaptations of a red blood cell for oxygen transport. Answer. Its biconcave shape gives a large surface area and short dFY-oxygen distance. Absence of a nucleus leaves more room for hemogloHbin, which binds oxygen. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. Practice. A cell image is 48 mm and magnification is x800. Calculate actual size in micrometers. Answer. 48 mm equals 48,000 micrometers, actual size equals 48,000, all over 800 equals 60 micrometers. Watch the numbers land in the matching symbols. Check that units cancel cleanly. If they do not, the setup is wrong even before the arithmetic. Okay, that's it for cell structure, organization and microscopy today. Next time in O-level biology, we'll get into movement of substances across membranes. Stay curious and see you in the next lesson.